Niacin's involvement in glucose metabolism is a complex and nuanced topic that underscores how one nutrient can exert both therapeutic and contradictory effects depending on context. On one hand, niacin is fundamentally involved in insulin signaling and glucose utilization at the cellular level, supporting the body's ability to manage blood sugar effectively. On the other hand, high-dose niacin therapy, particularly in the form of nicotinic acid, has been shown in some cases to increase insulin resistance or worsen glycemic control, especially in individuals with type 2 diabetes. This dual identity has led to confusion and hesitancy in using niacin in metabolic care, despite its deep biological involvement in the very systems that regulate glucose. At the molecular level, niacin contributes to glucose metabolism through its conversion to NAD+ and NADP+, which are essential cofactors in glycolysis, the Krebs cycle, and the pentose phosphate pathway. These pathways determine how glucose is metabolized, stored, or used for energy. NAD+ is required for key enzymes that extract energy from glucose, while NADP+ plays a role in producing the reducing power, NADPH, needed for biosynthesis and oxidative stress defense, both of which influence insulin sensitivity. More specifically, niacin impacts insulin signaling through sirtuins and other NAD+-dependent enzymes that regulate metabolic gene expression. These enzymes can enhance the function of glucose transporters like GLUT4, which are responsible for moving glucose from the bloodstream into muscle and fat cells. In experimental studies, niacin and its amide form, niacinamide, have been shown to improve insulin sensitivity by supporting mitochondrial function, reducing oxidative stress, and lowering inflammation, factors closely tied to insulin resistance. In these contexts, niacin acts as a metabolic enhancer that helps the body respond to insulin more efficiently. However, the narrative becomes more complicated when we consider the effects of high-dose niacin, particularly nicotinic acid, which has long been used to manage cholesterol. At therapeutic doses, 1 to 2 grams per day, nicotinic acid can lead to transient increases in fasting glucose and insulin levels. This effect appears to be dose-dependent and is likely related to niacin's influence on fatty acid metabolism. By suppressing the release of free fatty acids from adipose tissue, high-dose niacin may reduce insulin sensitivity in the short term, since elevated fatty acids are an important energy substrate and their abrupt reduction can alter how cells respond to insulin. Yet this effect is not universal. In other populations, particularly those with subclinical niacin deficiency or metabolic syndrome, moderate niacin supplementation may actually improve glucose regulation over time. Niacinamide, the non-flushing form, has been studied for its ability to preserve pancreatic SS cell function and delay the onset of type 1 diabetes in high-risk children, although results have been mixed. Some research also suggests that niacinamide can reduce oxidative stress and inflammation in insulin-producing cells, offering protection against diabetic complications. This contradictory profile has led to niacin being both praised and avoided in diabetes care. Clinicians often err on the side of caution, especially with patients who are already struggling to control blood glucose. As a result, niacin's broader metabolic benefits are often ignored or overshadowed by the fear of glycemic disruption, despite the fact that the negative effects are usually mild, reversible, and context-dependent. It's important to understand that not all niacin is created equal. Nicotinic acid, niacinamide, and inositol hexanocatenate all behave differently in the body, with distinct impacts on lipid metabolism, insulin signaling, and inflammation. This means that blanket statements about niacin's effects on glucose control can be misleading unless we specify the form, dose, and the individual's metabolic context. For example, using niacinamide at low to moderate doses may support insulin function without the glucose spikes associated with high-dose nicotinic acid therapy. In broader terms, niacin's influence on glucose metabolism reflects its central role in maintaining metabolic flexibility, the body's ability to switch efficiently between burning fat and glucose depending on energy demand. Metabolic inflexibility is a hallmark of insulin resistance and type 2 diabetes, and niacin's ability to support NAD+ levels, enhance mitochondrial efficiency, and reduce low-grade inflammation means it should be considered a key nutrient in restoring this balance. In conclusion, niacin is deeply embedded in the systems that govern glucose control and insulin sensitivity, but its narrative is muddied by dose-dependent effects and inconsistent communication in both research and clinical practice. Rather than dismissing niacin based on fears of elevated glucose levels, it would be more productive to refine our understanding of how and when to use it, tailoring its application based on individual metabolic needs, choosing the right form, and monitoring responses. Done wisely, this approach could restore niacin's rightful place in the toolkit of metabolic health and diabetes management.